Okay, all right. So thank you everybody for coming. Thank you very much to the organizers for inviting me. It's a pleasure to be back in India. I was here many, many years ago. I was working in CCMB in 2008 as part of my PhD, some of the work you heard about it. So I'm very excited to be back and to tell you about the work I've been doing since. The first thing you'll notice is that I completely changed my title and my topic. When I heard that I was speaking in an evolutionary session, especially after Ian Matheson, I told Ganeshwar, I said, what do you want me to talk about? My group in Australia, we have two very different lines, well, two complementary lines of research. Part of me is still very interested in natural selection and evolution, and part of me is doing more biomedical work. And a lot of that work is happening in Indonesia with Indonesian researchers. But today I thought I would talk to you instead about actually the work I've been doing on natural selection. And so I like to start my talks by acknowledging the people who were part of the work that I'm presenting, partly because I always run out of time at the end, and I think it's really important to make sure that we acknowledge their contributions. And so the work you'll hear about today was by some of the members in my lab, in particular Davide and Madi, but also in both collaboration with researchers at the University of Papua New Guinea, the University of Toulouse in France, Cambridge in the UK, Massey in New Zealand, and the University of Tartu in Estonia, where actually was the first time that I met Dr. Ganeshwar and Tandra. And so I'm very happy, actually, Ian sort of gave my introduction to me. I'm very grateful. So when we think about humans appear in Africa somewhere around 300,000 years ago. The numbers are always changing, but the overall pattern is the same. At some point, humans leave Africa, spread throughout the world, and that's kind of what we know about. That's like sort of the briefest version of human history that I can give you. But what I like is not so much humans, but what came before them. So if we go back in time a bit deeper, we find around 700,000 years ago, there was a split in the lineage that gave rise to two other archaic hominin taxa known as Neanderthals, who were very common in Eurasia and Western Eurasia, and Denisovans, who were very common in maybe East Asia, perhaps Southeast Asia. It's not quite, we don't know a lot about their geography because we have a very, very limited fossil record for Denisovans. What we do have, though, from those species in 2010, which was sort of the advent of ancient DNA technologies, is actually genome for both of these individuals, both of these taxa. And so we've been able throughout the years, and when I say we, I mean the field, to identify instances of gene flow from either Neanderthals or Denisovans into diverse human populations at different points in time. And the consequence of this gene flow has been that around everybody in this room, and certainly everybody of non-African ancestry, carries around 1 to 2% of Neanderthal DNA in their genome, and that 1 to 2% is different for each of us, but we do all have that. And then those of us that are some people certainly of East Asian, a bit of South Asian descent, carry a small fraction of ancient DNA, of introgress DNA from Denisovans, around 0.1%. But where this really peaks is in New Guinea, in Papua New Guinea, in the Asia Pacific, in a region that I'll refer to as Island Southeast Asia, which primarily covers sort of Indonesia, Papua New Guinea, the Philippines, etc. And in these populations, we find up to 4% of Denisovan DNA. So a question that I always had, and I think a lot of people have always had, is like, okay, well, you carry these variants, what are their functional consequences? Why have these variants survived 50,000 years in the genomes of these people that carry them, such that today we also carry them? So it's a lot easier to answer this question when we focus on Neanderthals. Neanderthal introgression into modern human populations has given rise to things such as certain HLA alleles that are now common in certain parts of the world. But actually, at a genome-wide level, we have a pretty good understanding of Neanderthal introgression. And so we know that Neanderthal introgression, SNPs that are Neanderthal and are circulating today, are currently associated with certain traits, sometimes things like obesity or your risk of depression, or actually also red hair and skin pigmentation. And essentially, this work has been on there's been very extensively done, very extensively mapped, such that we understand the Denisovans introgression. Sorry, Neanderthal introgression. We do not understand the Denisovans introgression nearly as well. And the reason for that is in this figure that many of you may be familiar with. So a lot of these studies that seek to understand Neanderthal introgression do it by looking at genome-wide selections, genome-wide association studies, genome-wide selection scans, and these studies have been performed primarily in the population shown in red here, which are, hey, where's the pointer? Europeans. There you go. So we have a lot of data on people of European genetic ancestry, and by a lot, I really mean a disproportionate amount, far too much, in fact. This is true at the DNA level, so if you want to look at genome and association studies, you are very, very easy to find selection studies that have, association studies that have been done in Europeans. It's a lot harder to find data from anyone else in the world, despite the fact that Europeans make up only around 15% of the global population. And then recently, leaving aside DNA and moving on to functional genomics, to RNA, to gene expression data, me and a colleague have found that it's the same in this kind of data set. When we look at the ancestry or the geography or the race reported in samples, in public RNA sequencing samples from either the SRA, which is a public database, when we just mined the literature, we read 600 papers randomly and tried to work out what samples have they used. We find that they're generally of European ancestry, or when they're papers from the US, they're generally described as being of white race. And we also find this in dbGaP, which is another repository of public RNA sequencing data. So we have a lot of data in a lot of different modalities from European individuals, and we have very, very little data from the rest of the world, including certainly India, which is represented here by a couple of data sets also only. And so if I want to know what the Denisova interaction does, I can't actually just look up my SNPs on a database because they're restricted to individuals who are not European, and we don't have any public data from these individuals. So we have to get a bit more creative and a bit more helpful. And so a couple of years ago, so what means is that we only have like very nice, limited individual stories of Denisovan interaction. So we have this one here, again, a couple of HLA alleles that come from different Denisovan sources. We have this beautiful paper, if you haven't read it, I recommend it. That shows that there's a couple of coding mutations in a gene called TNF-AIP3, which codes for a protein called A20, which is a negative regulator of NF-kappa B signaling and inflammation. And that SNP is really common, not only in this part of the world where the Denisovan integration is higher, but it's also spread beyond Lithuania and seems to help fine-tune immunity. What that means I'll get to later. But I think really the most famous example of the Denisovan interaction was the finding in 2014 that the SNPs that allow Tibetans to thrive at high altitude in the Himalayas, so 6,000 meters above sea level, were of Denisovan origin. And so here you go, this is all we know about Denisovan integration compared to our genome-wide understanding of Neanderthal integration. So this is disappointing to me on many levels, scientifically, ethically, in many, many levels. And so we thought, well, let's do something about it. So working with some collaborators, we took some published data. They had sequenced around 200 genomes from, again, the island Southeast Asia, so this part of the world here, including genomes from New Guinea Island, as well as a lot of other islands here in eastern Indonesia and western Indonesia. And they had identified, Guy Jacobs and colleagues had identified Denisovan integration throughout the genome. And so we took all of these SNPs, we did a lot of QC filtering, and we ended up with around 100,000 archaic SNPs that could be of either Denisovan or Neanderthal ancestry, and then a bunch of control SNPs that we also identified to compare them. And we asked, okay, well, we cannot look these up again on a genome-wide database because they're not going to be there because they are geographically restricted to this part of the world. And this part of the world, if you notice during Ian's talk, there was never any samples from this part of the world. We said, well, let's look at something a bit more sort of unbiased or neutral. So what we did was we intersected these SNPs with chromatin states from the Roma genomics data set. And for those of you that are not familiar with that, this is the idea that we can look through the genome in different cell types and say this part of the genome in this cell type is being transcribed. Okay, this is a gene that is active in an immune cell, for instance. And the next part of the genome, a little bit over there, is transcribed in alpha cells. It's probably a housekeeping gene. This part, another part of it there, is open chromatin, so it's available to the cell can see it, but it's not transcribed. And so we think this must be a regulatory element. So something that controls when we turn on a gene, or how much of that gene we turn, how many copies of mRNA we make, and so on and so forth. And so we asked, where in the genome do the Denisovan SNPs and the Neanderthal SNPs fall? And the acronyms are horrible, so I will explain them to you as we go along. But what we find here is that both Denisovan SNPs and Neanderthal SNPs are enriched in these sets here, which are these acronyms that if you're not familiar with data, you will never try to work out, but they're basically enriched, overrepresented in parts of the genome that are, this one here is actually repressed chromatin, so they're often seen in parts of the part of the genome that are repressed and inactive, but they're also seen here, the three green signals here are enhancers. So again, features parts of the genome that control when we turn a gene on, how much of a gene is transcribed into protein, that control protein levels, not protein sequence, the sequence is the same, but the relative abundance between individuals varies. And we said, that's interesting, okay, that's nice. But then the beauty of rote epigenomics is that we can ask, enhancers for what? For what cell type? Is it cardiac enhancer? Is it, I don't know, skin pigmentation? What is it? And what we found was that Denisovan and Genful SNPs were enriched for enhancers that are active in blood and T cells, so immunity. And then there is an additional signal in Denisovans, but not in Neanderthals over here, and B cells, so other component of the immune system. So we find a real excess of immune signals, of Neanderthal, of Denisovan SNPs in regions of the genome that are thought to be enhancers for immune function. And this gives us a very nice testable hypothesis to sort of follow up on. We know that, or we believe as a field and collectively, that immunity pathogens is one of the strongest selective pressures you can encounter, right? Like if you're sick and it kills you, that's it, it's over. There is no coming back from that. And so selection for pathogens, selection for resistance against pathogens, for fine-tuning the immune system is generally very strong. But also, especially in Southeast Asia and parts of the world, tropical places are full of pathogens. That's just the way that the ecology works. And so you have really a strong selective pressure on the mechanism here. And so we found, okay, that sounds great, but let's test it a little bit. So we tested, in the first paper, we just tested one. There is a couple of genes here, OAS2 and OAS3. These are immune genes. They are known to be targets of integration, and we have found a few SNPs that fell in enhancers that seem to be active in these genes. So we had a couple of them. Each of these, this plot is complicated, but basically we have found a bunch of genes, bunch of Denisovan SNPs, each of them here, that fell into transcription factor binding sites, so sites where we actually had a mechanism of action for a bunch of different genes. So NF-kappa B, MAS, TAL1, etc. And we said, okay, well, that's great. It's still computational. It's still a prediction. How many of these are actually, can any of these SNPs actually drive a difference? Do you see a difference between the two alleles in actual reporter gene activity? And so we knew that these genes were differentially expressed between Western individuals from Western Indonesia who have no Neanderthal, no very limited Denisovan integration, and Eastern Indonesia, so in red here, who have high Denisovan integration. So that was nice. That was a confirmation that at least we saw a difference in expression levels that associates with the ancestry. And then we had a couple of cell lines from Papua New Guinea that we had established from genetically Papuan individuals where we had the genotypes. And so we were able to test to verify that two of these SNPs showed very significant differences in expression when you test the Denisovan allele versus the modern allele. And there were the SNPs that impact this NF-kappa B2 binding motif. So we said, that's great, that's nice, how wonderful, the Denisovan integration has a role. This one SNP, right? We have a problem. We have 50,000, 60,000, 100,000 SNPs, and I don't have 50,000, 60,000 students to ask them each to do a single reporter gene assay and fix that problem, right? And so we said, okay, well, there has to be a better, more high-throughput way. So the second half of this talk now is work that's being done by a student in my lab called Maddie Comerford. And so what Maddie did was she went back to the data, she went back to the chromatin states, to the epigenome data, and she went back to the immune cells and said, let's identify every single SNP in this data set, in our data set from Matt Jacobs, that has a minor allele frequency, where the Denisovan allele has an allele frequency at least 0.15. I think that's a nice cutoff. And let's just identify them. And it has to be in an enhancer that is active in immune cells, and it has to have an allele frequency of about 0.50. And then we say, okay, here's the two alleles. What we do then is we synthesize an oligo, so just the DNA sequence, 170 base pairs. So we synthesize the 120 base pairs around our SNP here, so 85 that way, 85 base pairs that way, and then we have 15 base pairs here that we'll use for some cloning later on. And so we identify around 15,000 Denisovan SNPs that meet this criteria, around 11,000 Neanderthal SNPs that meet this criteria. We throw in some controls, different kinds of controls. We also throw in some QTLs that I will not talk about, and we ended up with 54,000 of these oligos that are now sitting in a little tube, and we say we can test all of these. We can basically do what's known as a MPRA, a massively parallel reporter assay. So the way MPRA works, for those of you who are not familiar with them, is actually a really elegant, really simple technique. If you know a classic reporter assay where you take a bit of DNA, you put it upstream of a minimal promoter and then downstream of the promoter there is GFP, you put that in a cell, the cell turns green, then you say, great, my introduced bit of DNA is functional. The cell is not green, then your bit of DNA is not functional. So MPRAs do basically the same. Here are oligo sequences, but we add a little modification here. The bit in black here are DNA barcodes. So each of our oligos is tagged with a 15 base pair DNA barcode so that I can sequence that barcode and then say, okay, I see this barcode, this came from this oligo, which matches to this particular sequence in my database, right? And so now we have a tube with plasmids where we have each of our 54,000 variants is tagged with a bunch of barcodes and we know which one it is. And what we can do is we transfect that into a cell line. We stick those 54,000 plasmids into cell lines. We wait 24 hours, extract the DNA and the RNA from that cell, and then we simply count how many times do we observe any of our barcodes. And the metric here is then the ratio of barcodes that came from the DNA, but because this barcode is downstream of a promoter, we can also count if it is transcribed into RNA. Transcribed. So we can get RNA abundance for each barcode and DNA abundance for each barcode. When those two numbers are basically the same, or the RNA abundance is lower than the DNA abundance, that means the sequence is not very active. But when the abundance of the RNA barcode is higher than the DNA barcode, that's when we say, this sequence that I tested has the potential to drive gene expression. And so we do that, and we get a bunch of results. So I will skip some of the math. Basically, what we end up is actually, remember we started with 15,000 Neanderthal sequences. We found around 10% of them showed some level of activity, so they were active, they were capable of driving gene expression. Around, again, 10%, 11% of Neanderthal sequences we tested were capable of driving gene expression. But we're interested not so much in this part of the genome is functional, which is what got up here. We're interested in instances where the Denisovan allele behaves differently from the human allele at the same site, right? These are the instances where the Denisovan integration could have actually had some sort of measurable functional impact on the individuals that carry the data. And so we have a pathetic little number. So only 93 cases of that in the Denisovan data. So 93 SNPs in the genome where the Denisovan allele drives measurably different expression than the modern human allele, and around 85 where the Neanderthal allele drives different expression levels from the modern human allele. And so here's a volcano plot of our data. If you count here, that's 138 points. If you count down here, if you want, that's 3,400 points. But the ones we liked, the ones we have focused on at first, are these two here, are very strong signals. So these strong signals come from two genes. The red one here is a Neanderthal signal. I don't like Neanderthal, so I'm going to skip it. The blue here is much more interesting. It's again TNFAIP3, which, if you remember, I told you is one of the only instances that we know of adaptive Denisovan integration in our data. So TNFAIP3, what is the gene doing? This is not our SNP, this is just the gene again. TNFAIP3, I told you, is a negative regulator of NF-kappa B. And so what it does is actually it fine-tunes the immune response, such that when you carry the Denisovan allele of this mutation, not our mutation, you actually have a slightly better response to pathogens. And so you're better able to clear some immune challenges than when you have the modern human allele. And so here is the pathway. TNFAIP3 of the protein is known as A20. It's a D-ubiquitinase, but it also is an ubiquitinase. It's a bit complicated. We're still trying to get our heads around it. But we say, okay, that's an interesting, again, it's a hypothesis we can test. How do we actually validate this in our data, right? And so the part of my life that I haven't told you about where I work in Indonesia, one thing we've been doing is we're doing a lot of single-cell RNA sequencing of whole blood from Indonesian individuals. And we've sampled two parts of the country. We've sampled Bali, so individuals that are genetically of Southeast Asian ancestry, and we've sampled Papua, which are individuals that are very similar to the ones that carry the Denisovan integration. And so we can ask, do we see different levels when we look for our SNP, the one in our NPRA? Do we see different levels of expression of the TNFAIP3 gene relative to genotype, yes or no? Because that would be a very nice confirmation that our SNP does something in cells. So we see some differences in CD4T positive cells. They go the wrong way though, so here is the alternative allele. We think our thing is an enhancer, and it seems to be going the wrong way, so it's a bit confusing. We also see the same trend in myeloid cells, again, lower expression in the homozygotes. That's a bit complicated, it's hard to interpret. When we segregate though, when we group individuals by ancestry, what we find actually is that the trends are very different depending on ancestry and environment, and so we're trying to disentangle that. But we do see, what we like to see is in B cells. So B cells are one of the immune cells, of course, they're very key. And here we find that when you have more copies of the SNP we identified, you do get higher expression of TNFAIP3, which is exactly what we would predict. And again, this sort of seems to be independent, though, here of genotype. And so it's still complicated, hard to understand what's happening, and it's hard to line it up with the findings of this paper. And so we're still very much trying to understand what is our SNP doing? Is it behaving in the same way that the other SNP that's been found? Or is it separate? Is it a totally different selective force? We know that they're not linked to each other, so it's not they're not simply hijacking each other. But this is sort of the sort of work that we can actually now do by combining our MPRA data with our gene expression data that we have generated from this part of the world. And so that's one SNP. I want to tell you though in the overall trend, so we have looked at more than one SNP. And so what we actually find is that if we look at the distributions of activity, I told you that anything where DNA was higher than RNA is inactive, uninteresting. So these two peaks here, they're very faint, this one and that one, are our sequences that we tested, and you can find that the vast majority of them are below 0. So most of them seem to do nothing at all, in fact, at least in the cell types that we use. This is different from our controls, so we do trust our assay. The positive controls did drive expression. The ones we tested did not drive expression. And you did. And so this is my last slide, don't worry. And so what we do find, though, is that there are slight differences in where those active SNPs fall if we look at the different ancestries, the Denisovan and the Neanderthal ancestry. So Denisovan SNPs that are active are very close to the transcription start site. They seem to be in promoters. This is not true for Neanderthal alleles. There is a big difference here. And Denisovan SNPs that are active are present in Papuans at very high allele frequencies, which is really suggestive to us of actually functional adaptive integration that has been maintained for 50,000 years. And so just to recap what I've told you, basically, Denisovan integration, but not Neanderthal integration, is overrepresented in immune cells in Papuan individuals. An MPRA that will test consequences of these SNPs and beyond simply computational analyses. And we do find measurable differences in the patterns of activity between Denisovan and Neanderthal variants. And then we have the single SNP that we're trying to understand. It is still very confusing to work out exactly what it is doing and whether it's a driver of selection. It's an independent selective disease, independently targeted by natural selection. And with that, I'll thank my lab again, this time the whole lab, and all of my other collaborators that I didn't get a chance to talk to. Thank you.
I'm sure there will be exciting photos. Just curious, that switch that you found, is it EQTL?
It's a great question. So we haven't finished testing EQTLs in our data yet, so I cannot tell you about our data. I think it will be when you look at some of the patterns. Like, this is basically artificial, artisanal EQTL testing. It's not present anywhere else in the world. So the allele frequency for our SNP is 0.65 for the alternate, so it's very high. So we definitely will have power, and I would say it will be a QTL. But it's not seen anywhere else in the world, so I can't look it up in GTEx or anywhere else. I would love to be able to do that.
Professor, WG tools use baseline allele frequencies to deploy. For the haploid, it's 1 N for diploid, 0.51 for triploid, 0.80, 0.67, and 1 for tetraploid, 0.25, 0.5, 0.7. So the cardinality of the ploid, like the tetra or tri, can it be abstracted using roots of unity? For example, a square root of unity minus I and I, cube root of unity or omega to the particle. Somehow correlate the roots of unity to these proportions.
So,
provide mathematics.
I'm not sure I got it.
The cardinality of the ploid is like tetraploid can be 4 roots of unity, and triploid, cube roots of unity, diploid, 2 square roots of unity. So for the cardinality of the roots of unity, can we somehow corroborate into the these frequencies, like for baseline allele frequency for haploid 1 and
so humans are always diploid, right? So except for our germ cells, everything is always diploid. This is population allele frequencies, and so basically, when you have enough observations, you're good to go. You can find anything, any enough individuals, any possible, any other frequency is possible within a single individual. It's either 0, 0.5, or 1. That's it.
Most trivial case, I think, because we are talking about it deployed in this case. So you see this.
I think it's a very nice talk
Thank you.
and compelling observation. I'm really interested in your enhanced repertoire. You and I roadmap it, states are derived from cell lines that exist for times, right? Whereas you had an opportunity to target the human cell axis. They have produced enormous amount of attacks, single cell attacks. Why I'm interested in that because I also compared immune cells which are immune, which you get from the culture cells. And then when you really compare it with the single cell axis from the frozen tissue, it's completely different.
Yeah.
Don't you think that would be still?
Oh, I would love to do that. So I mean, epigenome workup is mostly primary. It's a mixture. It's weird primaries, right? It's not just raw primaries. We also have been doing attack. We haven't analyzed it in our data, so we have attack data from 16 of these individuals. The other complaint that you can make if you want, and I think it's a very valid criticism, these are immune cells that are at resting state. They're not fighting an infection. So the other thing we've been doing, which I haven't shown you, is we've got our cell lines and we stimulate them in the lab with different pathogens, and then we measure gene expression to see, because I don't necessarily expect to see this SNP being active at baseline. It's a negative regulator. It should be doing nothing. NF-kappa B should not really be active in resting cells. So we actually do it in stimulated cells, and I'm still waiting for my students to submit a qPCR results, otherwise I would have put them in here. And that's a much more realistic test of what we're actually doing. Yeah.
And especially this evolution, it should be happening in response to the loss of pathogens.
When you see something, yeah, not at baseline. I agree completely.
It would make more sense to stimulate.
I think we have to. I think the last question here.
Great, Dr. Was there a period of comparing the bulk RNA seq data for your single cell RNA seq data?
Have we done that? So I don't think we've done that actually. We have a panel of around 100 individuals with bulk RNA-seq, and so they come from 3 sites in Indonesia. Western Indonesia around 50, Central Indonesia around another 50, and then 19 individuals from Papua. So we're really underpowered in Papua at the bulk level. But then at the single cell level, we have 100.
One final question: is there a plausible reason that there is a large amount of contamination over the years that could have predisposed the introgression to the non-coding regions or the regulatory regions? Could you also look into further open format elements looking beyond the CTCF motifs or further those kind of things?
So
is there any kind of progression that you can see?
We've been doing high C on the cell line, so again, we haven't finished this. One of the things we're very, very interested in is actually the CTCF motifs, the introgression require the chromatin architecture. Our preliminary result is that either we have no power to see that or that it's very rare, but we also know there are a lot of motifs, CTCF motifs that are in the introgress regions, and so we're still trying to work out how do we link different scales of genome regulation together, because I agree with you. It's a very obvious sort of next step. So yeah.
Thank you very much. It was a wonderful talk, and I'm sure you will enjoy it. So now we come to the third group of you by Dr. Irene.